Vitamin A. Yeah. So yes, student, it is one of the application of the biotechnology in the agriculture. So here the golden rice is consisting of the beta carotene gene. Okay. So that's why we will call it as the, so that's why it is the yellow in color. Okay. Beta carotene. You have know about the genes which is causes the pigmentations, right? So this is vitamin A. It is going to carry the beta carotene gene. Okay. Beta carotene gene. It is a reason for rice to look yellow color. Okay. So not the omega 3, not the vitamin B, and not the vitamin C also. Okay. Vitamin A. Beta carotene gene it is going to carry, and it is going to use for the disease. Can anyone tell me deficiency of the vitamin A will cause which disease? Deficiency of vitamin A will cause the which disease? I think you have studied in the chemistry also. Right? Can anyone tell me which disease is this going to be caused? Can anyone tell me which disease it is going to be caused? Student? Yeah. So night blindness, right? So it is going to cause the night blindness. So whenever the vitamin A is not there, it is going to cause the night blindness. Night blindness. Okay. So for the patient who is suffering from the night blindness, we will give this beta carotene. Okay. So that is the vitamin A we will give. So this is the very useful for the patient suffering from the night blindness. Okay. Yeah. So then we will go to the applications of biotechnology. Okay. So these are the biotechnology applications in the field of pharmaceutical, in the field of therapeutics and diagnostics and genetically modified crop, and also the processed food and the bio remediation, waste treatment, and the energy production. Okay. So if we start telling this means it will take the huge time. We are going to study only the applications which are given in your textbooks. Okay. But these all the applications. One is in the biopharmaceuticals and therapeutics, diagnostics, genetically modified crops, and processed food, and bioremediation. Okay, so these all the application in the application of biotechnology in all the fields. Okay, so actually, what is the main thing here we have to study? So the critical research areas in the biotechnology. See here, it provides the best catalyst in the form of improved organism, usually microbe or the pure enzyme, right? By the process of the recombinant DNA technology, it is possible to get the pure enzymes, right? So we can utilize the pure enzyme for the product formation, right? So these things will use in the industries. Okay, this thing will use in the industries. Okay. So, and the second thing is usually the microbe of the pure enzyme they will use. They create the optimal condition through the engineered for the catalyst to act. Okay. So here the catalyst is what? The biocatalyst. It is also the same enzyme. Okay. So it will create the biocatalyst, the enzyme they are going to produce. And you have studied about the bioreactors, right? The bioreactors, they are going to produce the bioreactors. In the bioreactors, it is possible to maintain the optimal temperature, optimal pressure, and get the desired product, right? So that is also the one of the thing. And downstream processing technologies to purify the protein and the organic compound. Whenever we produce the any product in the bioreactor means it is usually consisting of the so many contaminants, right? So by using the biotechnological process, downstream processing, we will recover only the desired product at the end of the production. So these all the things mainly involved in the biotechnology. Okay. So then let us study one by one. Okay, in the application in the agriculture. So I have a question for you students. So who is the father of green revolution? Can anyone tell me who is the father of green revolution? I need answer from you. Who is the father of Green Revolution? Okay. Oh, my slide is not changing. Okay. So now I'm student. Now I'm in the agriculture application of the agriculture. It is not visible to you. Can anyone tell me? JNV Shimla, can you tell me whether it is moving? No. Okay. Okay. Thank you. Wait, I will do one thing. Wait. Now it is moving. Can you answer me? Now it is moving? Yes. Okay. Thank you so much. Yes. So can you tell me, student, who is the father of Green Revolution? I think you have studied that in a health and disease. In the health and disease chapter, they are given the scientist name. Okay. Yeah. So the scientist name is M.S. Swaminathan. Okay. The scientist name is M.S. Swaminathan. M.S. Swaminathan. Okay, M.S. Swaminathan is the scientist who is given the Green Revolution. Okay, so he is used the method, so that is the agrochemical based method. Agrochemical based method. Agrochemical based method. Okay. Okay. So if we know that day by day the population was increasing, right? So we have to meet the food production, right? So can you tell me by which all the methods we can increase the food production? By which are the methods we can increase the food production? Can you tell me, students? By which are the method we can increase the food production? I need answer from you all. So the first method we can talk over the agrochemical based method. Agrochemical based method. Agrochemical based method. First one, and second one is the organic farming method. Organic farming method. Right? I have got an answer. Very good, very good. Genetically modification, right? So another method is the genetically modified method by the genetic engineering method. By genetic engineering method. Genetic engineering method, right? So by these three methods, we can increase the crop production, right? So one is the agrochemical method, organic farming, and another one is the genetic engineering method. See here, what we will do in the agrochemical method. So in the agrochemical method, so I have told you that M.S. Swaminathan sir has given that agrochemical based method. So is the reason for the Green Revolution, what happened on the time? So the food production was increased to the triple time. Okay, triple the food production was increased. 1 minute. Okay, so the agricultural product was increased to the triple, but here we are using the chemicals. Okay, in the agrochemical methods, we are using the pesticide, insecticides, and the chemicals. It will be going to be cause the environmental hazardous. Okay, so that is a method we can increase the food production, but the problem in the agrochemical method is, so here we are going to the agrochemical method. Agrochemical method is, so here we are using the fertilizers and pesticides we are using. So these all are the chemicals, right? These all are the chemicals which is going to produce the environmental hazards, right? Environmental hazards. So that is the one problem in the agrochemical method. And second environmental hazard is the first one. Second thing was, so it is very difficult to purchase the chemical is very costly. Okay, this fertilizers and pesticides will cost so much, so that is difficult for the farmers to buy this. Okay, so that is also the one more thing. I am changing the slide now. 1 minute. I am changing here. Have you know? 1 minute. 1 minute. My screen is not moving. Students, can you tell me my screen is not moving? Is it? Okay, 1 minute. Some network issues there, I think. Okay, 1 minute. Now it is moving. Students, now it is moving. Yes. Okay. Thank you so much. Yes. So the three methods is, one is the agrochemical based agriculture, second one is the organic agriculture, and genetically engineering based agriculture. So these all the three things we have studied, right? So in the agrochemical method, we are going to use the fertilizers and pesticides we are using here. Pesticides, right? So we can use this, but the main problem is, I have told you that environmental hazard is the first one. Environmental hazard is the first one, right? So the second one is it is costly, right? It is costly. The chemical will cost, right? So that is the thing. The second thing was organic agriculture. Okay, so in the organic agriculture, what we will do by using the cow dung and by using the organic manure, we are going to synthesize and we are using for the production of the things. But here the main problem was, we will not get the high yield. Okay, we will not get the very much high yield for the increasing population. So that is also the one more thing. So, but finally the left out thing was the genetically engineered based agriculture, right? So genetically engineered method we are using. So that is the genetically engineered plants we are using. So we can genetically modified organism by using that. So one is we can utilize the plant also, and we can genetically modify the plant also for the growing, for the product, for the good yield, or we can use the some bacteria or fungi and the animals, so for the manipulation. 1 minute. Okay, so this is the process. Okay, let us study one by one. Okay, so which are the advantages of the genetical modified plants? Students, can you tell me which are the application you know first? Okay, so I think this chapter was done in your school, or I don't know, in your college, or I don't know. Can you tell me which are the application you know in the agriculture? I will also help you. Also tell me which are the applications. Pest resistance. Can I tell pest resistance? My slide is moving, right? Pest resistance and insect resistance. Insect resistance, right? Pest and the insect resistance we can do. And second thing was the modified plants to tolerate the abiotic stress, right? So modification of the plant, modification of plants to tolerate to tolerate abiotic stress. To tolerate abiotic stress. Okay, abiotic stress. And third one is the post-harvestal loss. Post-harvestal loss. Can anyone tell me some other application in agriculture? Can anyone tell me, student, other application in the agriculture? Yeah. So disease resistant plant, right? Disease resistance. Disease resistance plant. Okay. So these all are the applications in the agriculture. So one is the pest resistance or the insect resistance we can make. And one is we can prepare the plant. So we can made the plant, genetically modified the plant to tolerate the abiotic stress. So that may be the temperature or the drought condition or the cold condition. So these all the things we can modify in the agriculture. So third one is the post-harvestal loss. Here you can take an example of flavor sour tomato. Okay, so here it is the genetically modified, that is the flavor sour tomato. Okay, so can anyone tell me which gene here it is they have modified? Can anyone tell me if you know that gene name? So here student, they have made they have made that one gene. So that is one enzyme to slowly release or slowly act. The name of that enzyme is polygalactourase. Okay, polygalactourase. Okay, polygalactourase. This enzyme is responsible for the tomato to ripen fast. Okay, so what they have did is they have released this enzyme very slowly. Okay, so or you can tell that functioning of this the enzyme becomes slow. Okay, because of this function, so the tomato can be ripened slowly. Okay, tomato can be ripened slowly and it can maintain its texture and it can maintain its flavor and all the content. Okay, so that is the main function of the polygalactourase. It is going to cause the ripening of ripening of tomato. Ripening of tomato. Okay, so due to this inhibition of this polygalactourase enzyme, we can make the tomato to be ripened slowly. So okay, so that is the one of the example for the post harvestal loss, right? So the farmers will cultivate this tomato and within a period of the time they have to sell that, right? Otherwise what will happen, it will be cause the loss to the farmers, right? Because this genetically modified tomato, so this post harvestal loss should be minimized. Okay, so this is the one thing. So let us study these all the things one by one. Okay. Okay. So, and the pest resistance plant. So what we are going to do in the biotechnology is, so we know that this particular plant was affected by this particular insect and particular pest, right? So we are introducing that gene into this modified plant, and that we are going to produce the pest resistant plant. So I have tell you that pest resistant plant created was the tobacco plant. I will give you the in detail of that while explaining that. Okay. One minute. My slide is moving, right? Can anyone tell me my slide is moving, right? Friend, can you tell me whether my slide is moving? Okay, 1 minute. Okay, yes. Yes, ma'am. Okay, thank you, Shimha student. Okay, so here see here, the pest resistants act as the biopesticides, right? So biopesticides, here you can see. And second one, they induce the needs for the insecticides. And the examples are BT cotton, BT corn, rice, tomato, potato, soya bean. So these all the plants we have made the pest resistance. Okay. Okay, so let us go through the pest resistant plant. Okay, so can you tell me what is the full form of BT cotton? I think you know, people. Can you tell me what is the full form of BT? Students, the BT means nothing but, I think all are studied before only, means they are, I think you know that. BC means that is a one of the bacterial name. Okay, Bacillus thuringiensis. Okay, Bacillus thuringiensis. Thuringiensis. So the main thing is this Bacillus thuringiensis bacteria will produce some type of the chemical. Okay, this bacteria is going to produce some type of the toxins. Okay, some type of the toxin that we will call it as endotoxin. Okay, endotoxin. It will be non-effective. Okay, it will not affect anything to bacteria, Bacillus thuringiensis. Okay, so that is in the inactive form. Inactive form. Okay, so whenever this toxin was taken up by the insect, okay, whenever this toxin was taken up by the insect, it is going to activate inside the insect gut. Okay, so whenever it is taken by feeding, whenever the insect was feed on this bacteria, it will not feed on. So we are taking the example of the plant. Okay, so whenever this taken by the insect, okay, whenever this toxin was taken by the insect, it will go to the midgut. Okay, it will go to the midgut. In the midgut, we know that the pH was alkaline, right? The pH was alkaline pH, right? Alkaline pH. In the alkaline pH, this is going to be activate. Okay, where student? In the alkaline pH, in the midgut, it is going to be activate. Then what happen? It is going to be bind on the, it is going to be bind to the midgut epithelial cell. Midgut epithelial cell, midgut epithelial cell. Midgut epithelial cell. Then what happen? It is going to create a pores. It is going to create a pores in cell. And what happen if the pores are created in the cell means the liquid things which is come inside of the cell, and it is going to cause the bursting of the cell, right? So bursting of the cell is going to be happen. Thereby it is going to cause the death of the insect, Okay, so this is the main function of this BT toxin. Okay, have you understood what is the BT toxin, student? Bacillus thuringiensis. It was released by the one of the bacteria, Bacillus thuringiensis. Okay, so whenever it is in this bacteria, it will not harm to the bacteria. Okay, because it is in the inactive form. So whenever it was taken up by the insect, in the insect midgut, there is the alkaline pH is there. In the alkaline pH, it is going to be activate, and so it is after the activation, it is going to bind the epithelial cells on the midgut, and it is going to damage the epithelial cells. Thereby, it is causing the death of the insect. So that is about the BT toxin. Okay. So now let us see how we are utilizing this BT toxin in the biotechnology application. Okay. See here. So this BT toxin is carried by the one of the gene. So that gene name we will call it as a cry gene. Okay. Cry gene is the name of the gene. Name of gene. Okay, so we will take this cry gene. So specifically, there are, here you can see here that gene was inserted into the cotton, right? So in the cotton, mainly the cotton plant is affected by the so many types of the insects. Okay, which are those insect means? So lepidopterans. Okay, lepidopterans is the first, and second one is the coleopterans. And third one is the dipterans. Okay, these all the types of the insect is going to cause the disease in the BT cotton. Okay, so can you tell me the example of lepidopterans, student? Can you give me the example of lepidopterans? So that is the birdworm and also the army worm, we will call it as. Okay, birdworm and the army worm. These worms, okay, these worms will comes under the lepidopterans, and the coleopterans are the beetles. Okay, beetles will comes under the coleopterans. Dipterans. Can you tell me the example of dipterans, student? It is very easy. Can you tell me the example of dipterans? Which are the things will comes under the dipterans? Our mosquito will come under the diphterans. Okay. Mosquito, and also the flies, house flies. Okay. This all the things will comes under the diphterans. Okay. So mainly the cotton is going to be damaged by these types of the insects. So lepidopterans, coleopterans, so and the diphterans. Okay, let us study. So how this cry gene was inserted. Okay, so students, mainly there are specific genes to be inserted for the specific crop. Okay, so for all the crops will not insert the same gene. For example, you can take cry 1AC, cry 1AC and the cry 2AB. Cry 1AC and the cry 2AB gene we will insert for the cotton. Okay, we will insert for the cotton, and the another gene is there. So that is the 1 minute. Cry 1AB. See here, don't confuse between the 1AB and the 2AB. Okay, 2AB is for cotton and the 1AB is for the, can anyone tell me? 1AB is for the corn. Okay, 1AB is for the corn. So these are the mainly specific genes we are inserted into the crops. Okay, into the crops. I think students have given the idea. Do you understood this? Can you tell me yes or no if you understood or not? Can you tell me whether you understood or not? Tell me yes or no. Okay, so let us start. Let us study about how this gene was inserted. Okay, 1 minute. Okay, see here what I have explained you here. Okay, so you know how this gene is going to be inserted. You know that, right? By the, for example, this is the cry gene. So here we are having the recombinant DNA, right? So here we are having the plasmid. So here we are having the plasmid here. So in the plasmid we are going to insert this cry gene, right? So this is, we call it as a recombinant DNA. Recombinant DNA. Recombinant DNA. So recombinant DNA, we know that by the Agrobacterium tumefaciens. Agrobacterium. By the Agrobacterium, we are inserting this into the plant. Inserting into the plant. Right? Now our gene was inserted into the plant now. Okay. So now what happened? Our plant is going to produce the cry gene. Okay. So now our plant is having this cry gene. So our plant is going to produce the cry protein. Cry protein, right? Can anyone tell me this cry protein is in the inactive form or in the active form? Yes, it is in the inactive form, right? So it is in the inactive form, right? So now what happened? This inactive protein was produced inside the plant. Okay. So now what happened? This insect will come. Okay, insect will come, or some pest will come, and it just go and bite on the leaves or any parts of the plant. Okay. Along with its saliva, it will take the Bt toxin. See here, along with this saliva, it is going to be inject here. Along with this saliva, it is going to take the Bt toxin. So see here, now it is going to the midgut. Okay, see here, solubilization and the activation. Activation has happened, right? Where? In the alkaline pH, right? In the alkaline pH. Can you tell me, student, what is the alkaline pH? Can anyone tell me what is the alkaline pH? In the pH, there will be acidic, basic, right? Basic means alkaline. Basic or alkaline, we will call it as light, right? So if it is below 7 means we will call it as acidic. If it is above 7 pH means we will call it as the alkaline or the basic, right? So this enzyme is going to activate only in the above 7 pH. Okay. So here now what happened? So this is, they are zooming this. Okay. See here, this what happened in this cell, they are going to explain here. Okay. So insect midgut. Okay. So in the insect midgut, this toxin is there. Okay. This toxin is going to be activated. Okay. So now what will happen? membrane insertion. Okay. Pores are lead to the osmotic cell burst. What happened? So once the activation was happened, so this is going to be activate. So where first what will happen? Here I will write the insect. Okay. Insect feeds on BT. Okay. Insect will feed on this. Then it is going to be activate. Where it is going to be activate? In the midgut. In the midgut it is going to be activate. So where it is going to bind? It is going to bind the epithelial cells of midgut. Okay, epithelial cells of midgut. So now what will happen? In the epithelial cells of the midgut, it is going to create some pores. Create pores. Okay, so just think what happened if the pores was created inside the cell. Okay, if it is a balloon, okay, if it is a balloon, if you created some pores in the balloon, what will happen, student? It will burst the balloon, right? So what will happen? So whenever the cell is there, see here, in the epithelial cell, what happened? Not only in the epithelial cell, in the immune system also, any damaging any cell, this is the mainly process. Okay, see here, the cell is there, it will create some pores in the cell. Okay. See here, it is going to produce some pores inside the cell. Okay, this is going to produce some pores inside the cell. What will happen? The plasma membrane was damaged, right? So whichever the solution or the liquid outside is there, so that will come inside the cell now. Okay, so this is the reason what happened. The inside, the liquid concentration is going to be increased. Then it is going to be burst out. Okay, so it is going to be burst out. The cell is going to be burst. Cell will be burst. Okay, so once the cell burst, what will happen? So is the one cell is burst? Okay. So all the cell is going to be burst and damage means what happens? It is going to cause the death. It is going to cause the death. So this is the main reason they will why they will insert this crygin inside the plant. Okay. I think you understood this. See here. So the pores are leads to the osmotic lysis. Okay. Osmotic lysis means nothing but it is going to cause the imbalance of the salt and the water condition. Okay. Osmotic is nothing but imbalance of the salt and water condition. So thereby it is going to burst out and it is going to be cause the death, cell death. Okay. So I think you understood this. Can you tell me whether you understood or not? Otherwise, I will explain you again. So JNV Shimla, Praveen, can you tell me whether you understood or not? This BT cotton. Yes. Thank you. Yes. So the same thing. Okay. For the BT cotton, what they are going to use here? Can you tell me which gene they are going to use for the cotton and which gene they are going to use for the corn? So the gene is, I have told you before also, the gene is the Cry1AC and the Cry2AB. 2AB, so that is for the cotton they are using. See here, this is the cotton. And for the Cry1AB, Cry1AB, okay, so they are using for the corn. Okay, if we want our corn to be insect resistance means I am adding the Cry1AB in the corn. If I want for my cotton, I want to produce the BT cotton means the gene should be in the BT cotton was Cry1AC and the Cry2AB. This is a multiple choice question. They may ask you. So just remember this. Okay. Okay. So I think you cleared about the BT toxin, I think. Okay. Let us go to the pest resistance plant. Okay. So here you can take the nematode. Okay. So nematode is the parasite. Okay. Parasite. You know what is a parasite, right? Parasite will always be heterotrophic. So it is like it is dependent on the nutrition for the other organism, right? So it is depend on the nutrition. It is depend nutrition on other organism, right? See here, they have created the pest resistant tobacco plant. Okay, see here, this is the Melodigne incognita. Okay, so this pest is the parasite or the pest is the nematode, the name is the Melodigne incognita. Okay, Melodigne incognita. So what it is going to do is mainly it is going to affect the roots of the tobacco plant. Okay, it is going to infect the roots of tobacco plant, thereby causing the low yield. Okay, so thereby it is causing the low yield. Okay, so the biotechnological application used in this method is the RNA interference. Okay, RNA interference, RNA interference, or we can call it as the RNA interference. Okay, so this RNA interference is the one type of the cellular defense which will take place only in the eukaryotic cell. Okay, this is one type of the cellular defense in eukaryotes. Okay, only in the eukaryotes we can see the RNA interference, not in any bacteria or any lower organisms. Okay, so then what will happen by taking this principle of the RNA interference? So what they have did is they have inserted the nematode specific gene inside the plant. Okay, by the method you know how they will insert this, right? By the Agrobacterium tumefaciens. First they have took the specific gene, the main gene which is responsible for the survival of this nematode. Okay, the main gene, that is the nematode specific gene. Nematode specific gene they have taken. Nematode specific gene. Okay, so nematode specific gene they have taken and they have inserted this nematode specific gene inside the plant. Okay, they have inserted this into the plant. You know by which method they have inserted, right? So that is the Agrobacterium method. Okay, so how they are inserted means this DNA is going to produce both antisense and sense RNA. Okay, both antisense and the sense RNA is going to produce inside the plant cell. Okay, so now what will happen? See here. So now our nematode is there, right? Nematode is going to affect, right? Whenever it is releases the mRNA, see here, nematode releases the mRNA in the cell. Okay, so now what will happen? So our RNA is there, right? Our antisense RNA and the sense RNA is there, right? See here. So mRNA is a single stranded. So that is by the nematode. Our RNA is also there. Our RNA, so that is there. See here, our RNA is there. So what will happen now? This RNA is complementary to this RNA. So what will happen? It is going to produce double-stranded RNA. It is going to produce double-stranded RNA. If once the double-stranded RNA is formed means, can anyone tell me the protein is going to be formed? Is that protein is going to form by the double-stranded RNA? Is it possible? No, right? Not at all possible. So this double-stranded RNA is going to be damaged. Okay, it is going to be broken by the cellular defense mechanism. Okay, it is going to be broken. It is going to be broken down by the cell. So our eukaryotic cell will not allow any double-stranded DNA, double-stranded RNA. Okay, not DNA, RNA. So that is the reason this double-stranded RNA is going to be broken down by the cell. Okay, so that's why. So whenever these important proteins mRNA are synthesized by the nematode, so the RNA, the DNA which is present in the plant body, it is going to produce the RNA. So this RNA is complementary to the nematode specific mRNA. Both the thing is going to be joined and the double-stranded RNA is going to form and protein is not going to be synthesized, right? So thereby, so this mRNA is going to be degraded finally, right? So finally the nematode is going to be die. Okay, so that is the reason they will use this mRNA interference, RNA interference. Do you have any question here? How they will do the pest resistance plant? So that is by the nematode. JNV Shimla, do you have any questions here? Can anyone tell me? Why? So that is the nematode specific gene. Without that gene, the nematode will not survive. Okay, that gene only they will insert into the plant. Whenever, obviously we know that, right? So by the transcription, mRNA is going to be synthesized. In the plant, mRNA is also synthesized, and this nematode is also going to be synthesized the mRNA. Both the things are complementary. They are going to be bind. So then protein is not going to be produced, right? So this is the main reason in the RNA interference. Okay. Okay, see here, they are given this. So this is the nematode infected tobacco. Okay, so whenever it was infected by the tobacco, here you can see here, this root, how it is, right? So this is the nematode infected tobacco root. So by this only, it is going to be produce the low yield in the crop. Okay, low yield. So this is the main thing. Okay, so then here is a question for you, student. In BT cotton, BT toxin present in the plant tissue. I have told you, in the leaves or in any tissue. Okay. As the prototoxin is converted into the active toxin. So whenever the insect bites this, the prototoxin is going to convert it into the active toxin. Okay. The option is option A, action of the gut microorganism. B, presence of the conversion factors in the insect. Option C, alkaline pH of the insect gut. Option D, acidic pH of the insect. Very good, very good. Yes. So it is due to the alkaline pH of the insect gut. Right. Yes, the alkaline pH of the insect gut is the correct answer. Okay. So let us study about what are the applications of the biotechnology in the medicine. Okay. So before going to that, I will tell you some other applications also. So have you know about the golden rice, right? Have you know about the golden rice, right? In the golden rice, what they will do, they are going to insert some in the normal rice. Okay, in the normal rice, they are going to insert some gene. So that is going to be carry the beta carotene gene. Okay, student, beta carotene gene. You know, beta carotene is the pigment. It is going to produce the yellow color pigment, right? Due to this beta carotene, it is going to cause the yellow color. So that's why we'll call it as it is the golden rice, golden color rice. Okay, so this thing we will give for this rice, this golden rice which is produced. Beta carotene is it is going to be inside. The gene is beta carotene. Then it is going to give the provitamin, which is that provitamin. So that provitamin is the vitamin A, vitamin A. You know that. So what is the deficiency of the vitamin A? What is the deficiency of the vitamin A, student? Night blindness, right? Night blindness. It is going to cause the night blindness, deficiency of the vitamin A. So treat that deficiency, treat that night blindness. The patients are going to give for the patients, we are going to give the golden rice. Okay. So vitamin A. So this thing you have to remember. Okay. And another thing I have told you that for the tomato, we are going to use the polygalactourase enzyme. I have told you, right? Polygalactourase enzyme we are going to use. We are going to release the slow enzyme. So thereby we are utilizing for the post-harvestal loss. Okay, minimizing the post-harvestal loss. So these are the main applications. In the textbook, they are given the one is the BT cotton and second one is the pest resistance. Those all the things you have to remember. Okay, so now let us come to the application in the medicines. See here. So the recombinant DNA technology helps in the synthesis of the therapeutic drug. Okay, the drug which is used in the therapies. And second one is so that only they are given. So that is the 30 recombinant therapeutic proteins are synthesized till now. So now 12 are working. Okay. See here, the first application is in the insulin production, genetically engineered insulin production. And second one is the gene therapy. And third one is the molecular diagnosis. So what is the main function in the gene therapy, in the insulin production, students? So many patients are suffering from the diabetes, right? So many people are suffering from the diabetes. So that is due to the non-proper or not synthesis of the insulin, right? Not proper synthesis of the insulin. So by that, what we will do? We'll synthesize the insulin and we will treat the patient with the insulin. So that is the one thing. Second thing was the gene therapy. So what is the gene therapy? We know that some genetic defect will be there in the people, right? So that will be from the by birth. So if some gene defect is there means, so that we can replace by the functional gene. Okay, so defective gene can be replaced by the functional gene. So that is by the gene therapy. Okay, that we are going to utilize the somatic gene therapy. There are two types of the gene therapy. Somatic gene therapy we are utilizing here. Third one is the molecular diagnosis. So nowadays you have know that if the person is having the corona means the which test they will tell to make, which test they will do? RT-PCR, right? Yes. The RT-PCR is the diagnosis test for the corona, and some other PCR test will be do for the diagnosis of the AIDS and for the cancer, right? And ELISA test will be do for the cancer. So these all the things will be the application of the biotechnology. Okay. So let us study one by one. See here, so genetically modified insulin. So first thing, student, you have to remember here. So what is this insulin? Okay, we know that this insulin is a protein, or we can call it as this is the peptide. Okay, so we have 46 number of the chromosome, right? So this insulin gene is going to be present on chromosome number 11. Okay, chromosome number 11. Okay, so see here, this is insulin is going to be produced by beta pancreatic cell. Beta pancreatic cell. In the pancreas, it is going to be produced. Okay, so that we will tell the beta cells of Langerhans, Langerhans cells. Okay, Langerhans cells. Okay, so see here. So how this genetically modified insulin is produced? Okay, so before by utilization of the biotechnological application, so the insulin was produced in some animals and pigs. They have utilized it and they are going to give for the patients. But what happened? The patient is used to show some allergic reaction towards that insulin produced by the animals. Thereby, so they have stopped that and they have started to utilize the biotechnological process for production of the insulin. Okay, so that's why see here how they have did is, first they have taken the human cell. Okay, so then they have taken the chromosome number 11. See here, they have inserted there and they have taken the bacteria. From the bacteria, what we will take? You know what we will take, student? We will take the plasmid from the bacteria, right? And this is the gene which is coding for the insulin. This is the gene which is coding for the insulin. So we have taken the gene from the human and we have taken the plasmid from the bacteria, and we will insert these both the things and we will make the recombinant DNA, right? So see here. So this gene is taken, this was inserted here, this is the plasmid, then recombinant DNA is produced, right? Recombinant DNA is produced. This vector is carrying our DNA now, right? So now it was inserted into the bacteria. So now this bacteria is going to produce the gene and it is going to be produce the proteins. Then that proteins is utilized for the insulin production. Okay. So how this is? It is not at all an easy process. Okay, because the bacteria is having some other gene regulation or the gene processing, and humans is having some other gene processing. So because you know that, right? Introns are not present in the bacteria, whereas it is present in the humans, eukaryotes, right? So that's why it made difficult to produce the insulin protein in the bacteria. So let us first study the structure of the bacteria, structure of the insulin. Okay. So first let us study the mature insulin. Okay. So mature insulin structure is like this. Okay. It is made up of the two peptide. One we will call it as the A peptide and another one we will call it as the B peptide. B peptide. Okay. So this A peptide is made up of the 21 amino acid and B peptide is made up of the 30 amino acid. Okay. Between these two, there will be the disulfide bond. Between the A chain and the B chain, there will be the disulfide bond. and there is the intra-disulfide bond is also present in the A chain. Okay, so see here, this the A chain is only having, right? This A chain is only having the disulfide bond. So that's why we'll call it as the intra-disulfide bond. So between A and B chain is the disulfide bond. So that's why we'll call it as the inter-disulfide bond. Okay, so let us see here. A peptide and the B peptide. So it is made up of 21 amino acid, 30 amino acid B chain, and between these two there is a disulfide bond is there. So there is a mature insulin. But the problem is this insulin was produced inside the body. It is in the pro-insulin form. Okay, pro-insulin form. Okay, pro-insulin form means it is not the mature one. This pro-insulin form, it is consisting of the three chain. Okay, three chain. One is A chain, and another one is the B chain, and another one is the C chain. But we know that in the mature insulin, there is no C chain, right? So then it is not possible to remove that C chain in the bacteria. So what they have did is, see here, this is the chain, right? So this from here to here, it is the C chain. So here they are given the mature insulin. Okay, this is the mature insulin. Okay, so here you can see this is the A chain. So this is the B chain. So whenever the mature insulin was formed, here you can see it is made up of only A chain and only B chain. So C chain was removed, right? C chain was removed. Okay, so how this A chain and B chain was removed? These all the things we will study in the next class. Okay, in the tomorrow's class, we'll completely study this all the things, how the insulin and all the things was produced. So do you have any questions here? Means you can ask me. Okay, student? Or I will go for the NPS poll. You can give me your feedback. Yeah. Thank you so much, students. Bye.